Assalamualaikum warahmatullahi wabarakatuh. This is our slide presentation for assignment 2, which is a specific interest of Pichia Pastoris and its significant application across various fields. Then, let me introduce our group members. They are Suhaila, Farah Amalin, Wafi Marina, Hakimi, Adlina Safia and Humaira Fashaha.
Application for insulin production. In medical field, human insulin is produced as recombinant protein using major route that involve the utilization of yeast-based expression system, leading to the secretion of a soluble insulin precursor into the culture supernatant. This route is economically viable. The metallotrophic yeast Pichia Pastoris has emerged as a very useful expression host with superior feature.
As for the advantages, Pichia Pastoris, like other eukaryotes, offers several advantages including proper protein folding, protein processing, and post-translational modification. In contrast, like Ecology, as a prokaryotic organism, lack the necessary folding machinery for complex eukaryotic proteins and cannot form the sulfide bond efficiently in its cytoplasm. Lack of this ability can limit the functionality of the insulin it produces.
Beside high expression levels, Pichia Pastoris has another significant advantage in glycosylation pattern of heterologous protein in comparison to Saccharomyces cerevisiae. It has been shown that heterologous protein in Pichia are not hyperglycosylated as length of the oligosaccharide chains added to protein is around 8 until 40 minus residues per side chain in Pichia, which is very short as compared to that in S. cerevisiae that is approximately -50 to 150 minus residues per side chain. Moreover, unlike in Pichia, glycosylated protein produced from S. cerevisiae have terminal alpha-1,3 glycal linkage, which are responsible for the hyperantigenic property of these proteins, and thus rendering them unsuitable for therapeutic applications in humans.
So now, this is process for insulin production in Pichia Pastoris. Step 1. Gene cloning. The human insulin gene is inserted into a plasmid vector, usually controlled by the maternal inducible AOX1 promoter and introduced into Pichia Pastoris cells. Step 2. Transformation and selection. The recombinant plasmid is introduced into Pichia Pastoris cell via transformation and cells are selected using antibiotic resistance marker. Step 3. Fermentation. The transformed cells are cultured in a bioreactor first using glycerol for growth, then methanol to induce expression of the insulin precursor.
Step 4 of the process for insulin production in Pichia Pastoris is protein expression and secretion. The insulin precursor is secreted into the culture medium, leveraging Pichia Pastoris' high cell density and secretion capabilities. After that, step number 5 is purification. The insulin precursor is purified from the culture medium using techniques like tangential flow filtration and cation exchange chromatography. Step number 6 is conversion to mature insulin. The precursor is enzymatically processed to remove the C-peptide and yielding active insulin. And the step last of the process is quality control. The final insulin product undergoes rigorous testing to ensure purity, potency, and safety.
In conclusion, the production of insulin using Pichia Pastoris as a recombinant protein involves inserting the human insulin gene into Pichia Pastoris cells through genetic engineering. The cells are transformed with a plasmid containing the gene, and positive colonies are selected and cultivated under conditions that induce insulin production. The yeast secretes the insulin into the culture medium, which is then purified through various techniques. And this method is effective due to the high growth rate of Pichia Pastoris and its ability to produce large quantities of protein with post-translational modifications. The purified insulin is processed as needed to ensure it is active and safe for therapeutic use.
Thank you for watching. Bye bye.